comment I had. And as far as honestly, I don't have much to say. Like
you just went through very quickly.
No. I read it properly. I can probably answer questions. I just don't know what to say.
Okay, Catherine.
Yeah,
I just see you doing this.
Yeah, because I one of the comments that Anish made on the article I replied to, it was about this like the age that they chose, and I was kind of wondering where, because they said the individuals who, like the sons that were involved, had to be 18 years and 9 months or older. So I was wondering where that specific cutoff was coming from, like why 18 and 9. It was just so specific, so I was confused, like why specifically, because a lot can happen.
The 18 should be easy to know because it's adulthood.
Yeah.
Right. So they want them to be adults so they can have consent to every part of the assessment.
Yeah.
And the 9 months, I guess, is just counting for the gestational period of 9 months. Okay. So they want to have that in place.
Yeah, it makes sense. I actually really liked the article because I last semester I interned at a gynecology clinic here in Sudbury, and they do run like a fertility clinic, and they do some sperm analysis and stuff for couples who are infertile. So they do look at the male component as well. So it was nice to see kind of the science side of it, just why, like the more specific stuff. I kind of saw it visually, but kind of seeing how they apply it too. There was two things that I was kind of confused as to why it was even included in the results section, because it seemed to be more cause and effect than like or correlation instead of cause and effect. And they were like totally, to me, out of place. The first one was they correlated the days of abstinence to the smoking status of the parents. So they said that days of abstinence were shorter in sons of smoking fathers than non-smoking. I didn't know why that correlation was even relevant to the study. Like, why does period of abstinence make a difference here as like relative to the smoking status? And the second one was spillage of sperm samples. And to me, I feel like that's literally, they were considering like that the sample was spilled but then they correlated that to the smoking status of the parents as well. So I was confused why.
There are a couple of things that they don't explain in the paper.
Yeah.
In the spillage is just because they are also evaluating the amount that has been ejaculated. So if it was spilled somewhere, the amount that they only did one sample, and that's a big no-no. You gotta have more than one sample that you can repeat just to assess the overall. Is ejaculation amount all the time equally is correlated with when was the last time of abstinence or masturbation, whatever type it is, right? So that is not, maybe they were trying to make another type of, this is one paper, right? We can use data in so many different ways. Maybe looking into, do people that smoke have more intercourse, or do people that smoke masturbate more? I don't know. We don't know that, right? So we don't know what other type of information these researchers were trying to obtain. So we just get limited to this. Maher? Maher, you have something to say about this paper? Did you read this paper?
No, I read the other article about the male infertility.
Okay, so
but there was an interesting part which I can link to the semen quality and this effect by this part where I read about the. Just give me a moment. Where I might use it as a link of the effect on the semen quality and the semen itself.
Yeah, because the other paper also talks about cigarette smoke.
Yeah, it's called because the stress induced by smoking, by psychological stress. But I consider smoking as a type of stress that can form on the body. where
Yes.
it induces alterations in the glucocorticoid actions on the Leydig cells, including the suppression of the androgen synthesis and induction of apoptosis. And
Yes.
apoptosis is controlled or automatic cell death. And it also has an effect through the 11-beta hydroxysteroid dehydrogenase mediated glucocorticoid synthesis, which inhibits the activities of the steroidogenic enzymes and thus impairs the steroidogenesis. So in other words, I think that this contributes to the low quality or the quality of the semen itself through these kind of processes. I don't know if I'm right or not, or wrong, but you are the one who can correct.
Well, there are many articles, and this is something that is very different that we have read in the past with Amanda for this course on the effect of the cigarette smoke. But it has a lot of issues. So yes, it activates your nervous system, and then it puts you in a more alert type of scenario, right? And that will alter your glucocorticoids. And if your glucocorticoids are altered, then your NRH hormone gonna also affect it, which affects directly the Leydig cells, which produce the testosterone, and it's a positive feedback, right, on that end.
If I may add on Maher's comment, most of the smokers that I know use smoking as a coping mechanism to stress. It's like basically a meditation for a few minutes, go outside, watch the street, and smoke is just an excuse. And the first paper touches that stress conditions can negatively affect the sperm count and sperm motility. So that might be another factor that I haven't really seen being considered associated with smoking.
Mm-hmm. Annika, do you have anything to comment on this paper?
I read the other one actually, so.
Okay, so just like I want to spare you for that.
Yeah,
Robert?
later maybe.
I read the other one as well, but
Okay.
I think a comment just on what Helen was saying is that, yeah, a lot of these factors kind of correlate. Alcoholism, chain smoking can all be related to stress in some way, and or overeating. A lot of these factors that would cause infertility can be related back to stress. So it's a really complicated sort of development.
Yeah. So, Luis, you put in the chat that smoking induces oxidative stress in the testes. Do you want to develop a little of that one, please? Mm-hmm. 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 Yeah. So I always put this type of papers that comes like a shock, right? And that's what I want to create on you. A shock, like, oh my God, once is to relate the material that we see in lecture and see how we can apply that knowledge, right? Because yeah, it's cool to talk about sperm and activation and how they fertilize the egg, but then it's what do we do with that information? And this is what we do with that information. This is how we apply. As I say, yesterday, it seems so far away with all things happening, that when we talk about fertility and the right definition of infertility is made in the other paper, nor in this cigarette smoke paper, we have this tendency to blame the female, right? If a couple is unable to conceive, immediately, what is wrong with her, right? And we almost never think about the male component into the infertility of a couple, right? There's this tendency of some males, but if I ejaculate, I'm fine, right? I produce every time that I have an intercourse, I can ejaculate. I don't have penile problems. I can get an erection with no problem. I get aroused. All that, and those are things that make the males believe that they are okay, right? But then there's a lot of people that are doing all this, but then they produce an ejaculation, but there is no sperm. They are oligospermic, right? They don't know until they look the slide underneath the microscope and say, oh, geez, there is nothing in there, right? Just like water. Jenna, we're going to go back to that comment of yours when we get into the next article, okay? But this is, as they say, that is a good 50% chances that the cause of infertility is male-related. Now, as you saw in both papers, for those that read both, there is a certain percentage of that we don't know why, right? So this paper is good just to show some of those direct effects of cigarette smoke. For those that smoke but don't want to say it, try to quit is better. Particularly if you want to have babies at one point. If you don't, hey, do whatever you want, right? But just consider, it's better to take a decision with information than just to deny reality, right? So you may be the cause of, not for you, but think about your kids. Do you want your kids also to have? Because there is a lot of other papers, and here they mention it, but they didn't give enough urgency to it, and it's a lot of the DNA demethylation that occurs, and that is epigenetic, and it goes up to the 4th generation, and it has been proven in many other studies. So it's not just you, it's your grandchild that can have some issues. And this goes, of course, part for the females. So it's just not part of the male responsibility, but also for the female, because, it gets a lot of effects along the line. Okay. Second paper, and I know that Emma read it because she had some questions. So go ahead, Emma.
I found the first paper on male infertility really, really interesting. There are a couple different aspects that I liked about it a lot. Also, thank you for sharing the article about the obesity and insulin. That was pretty interesting to read. I wanted to know a little bit more about that aspect of things. I really liked how they clearly defined what infertility was. And it's trying having intercourse 3 times a week for 12 months, but still not being able to conceive. So I personally myself, I didn't know that that's how long it was when you're looking at fertility and infertility when you're going through all these testings. And I know that a lot of couples experience this, and it's not something that's super uncommon, but I didn't.
We don't talk about it.
Exactly.
Nobody wants to talk about it.
Exactly.
Even if they are being trying to conceive and can get pregnant, they say, oh, we're taking our time, because it has a taboo. It's shameful. It's so stupid, this concept that we all have, right?
Yeah. And my aunt and uncle actually went through the same thing too, and they had no problem conceiving their first child, but it took them 5 years, and they were going through treatment and everything. But when we kind of knew that they were trying and they announced it, but nothing, like we knew that they were having trouble conceiving, it was the exact same thing. Oh, we're just taking our time. We're just waiting. So it was kind of like, not really wanting to talk about it. But what I thought was really interesting is that in a lot of these cases, as we've seen in both papers, there are a lot of different contributing factors to male infertility. So sometimes, and especially like the comment that Catherine just posted when they consulted that couple, and he said that he smoked a lot of marijuana in his teenage years. So it's pretty interesting because a lot of times I think couples don't think about sometimes the impacts that they've made to their bodies from past decisions. So I think sometimes that doesn't always come into play when it's happening in the moment. So I thought that was pretty interesting to read about how all these different lifestyle changes or things you do can influence so many years down the line.
Yeah. Annika? Thank you, Emma.
So what I thought about the paper, I like that it talked about like all the different things that might influence it, but what I thought was maybe missing from the paper was that they didn't like break down all the different variables to know exactly what the effect of just smoking is on that, or just obesity is on that. So like, for example, they talk about like phones and laptops, and someone commented that, oh, like, I wonder, since phones and laptops became so popular, if the rate of fertility went down. Okay, yeah, that's a good question, but since these things have become popular, there's more obesity, diets are worse. So like all that stuff kind of came at the same time with the new like less exercise and that new like lifestyle of online stuff and everything. So I think it would be interesting to like break down those different variables and see like, okay, like between just males that are obese and not obese and they both don't smoke and they both don't drink and something like that, instead of like just attributing it to one variable, but you're not looking at the rest. So how can you know it's not obesity and alcohol that's working together, like stuff like that? So anyways, that's what I thought was kind of missing from the paper.
Well, trying to recreate this type of experiments as we all think it should be is so fucking hard.
Yeah.
Aren't we exposed to everything and anything, right? So what about the CO from the cars, right? That will affect, and we are all exposed to it. Secondhand smoke. There are many studies that shows that secondhand smoke also can continue contributing to these effects, right? It talks about obesity, and yes, you're right. We are getting in an era where we do less exercise. We are more sedentary. We all able and capable of finding really good excuses not to go and hit 30 minutes exercise per day. Or our coping mechanism make us to eat cookies. My husband is buying cookies for my mom because he doesn't eat sugar. So it's for the kids, my mom that is with us, and I just don't buy them. I take one of those Maria packages of cookies and I sit in front of the computer and just like shovel, and I don't have a sweet tooth. But these cookies are horrible. I can't stop eating them. So I just want to need to take one and hide that package somewhere really far that I need to walk to get them, you know. And we are exposed to so many things. Makeup has a huge quantity of products based on petroleum, which is toxic for our body. I may show you guys a video later on, more towards the end of the semester, where we talk about these cosmetics and a latitudinal study where females were allowed to use their natural creams for hydrating the skin and things like that. And then they measured the level of those products in the baby. It was a mind-blowing thing to see how much toxicants of this has passed through the placenta to the baby. So we are exposed to so many things, but I don't want to talk too much. Robert and then Annalie. Robert?
Oh, sorry. Yeah, so I just saw the link that Sarah posted in the chat there. I was interested how they made the rats smoke e-cigarette vapor, and they basically just put them in cages with the vapor. Interestingly, they're doing. I googled it, and it is a lot of vapor. It's about like more than the average e-cigarette would have for a human person, and then giving that to a rat over a 24-hour period might be a little bit extreme. But I think it goes to show that anything in excess is going to play a negative role, no matter what it is.
But you say young. Yeah. Do you know the age at which kids are vaping now?
Yeah, pretty young.
More than young. More than young. So they're exposing themselves to all these products that have so many toxicants, right?
Yeah, and it's probably playing a different role in the developing body as compared to like an adult.
Yeah.
That role might be exacerbated, right?
Annalie, and then Kennedy.
There's a few things that I found actually pretty cool about these articles. I mostly read the infertility, the lifestyle one. I didn't know that a male had so much in part to infertility. Like I knew that they had some aspect to it, but I didn't know it was that much of an impact, including the different forms and shapes that the sperm can also have a huge impact on the fertility. What I found interesting is just taking off of what you said, Dr. Martinez, is that lifestyle is completely different now. And just because of that, it changes everything. Because beforehand, I know males were more active and very more about like their, work. And it was very, very physical, which has an impact on their sperm count, which is what it seems like. Also like radiation and stuff like that around us plays a huge part in the infertility, which some of it I believe we can't exactly control in some cases. What I found also very interesting is that infertility could cause diseases also. I believe the paper stated cardiovascular diseases, metabolic, oncological, and autoimmune systems. And I had no clue about that.
Okay. Kennedy?
One of the main things that I liked with this article was when they talked at the beginning about like how so much has changed in the past 50 years for male fertility. And like throughout the article, obviously they talk about how much has changed in the last 50 years. But like when you think about our diets, especially like the food that we're eating and all the processed foods and chemicals and contaminants that are in our foods, like I think that's a big thing. And they kind of talked about that, but not a lot. Like in terms of the foods that we're eating, they talked about obesity and that, but like I feel like that could definitely be something that they could touch on a little bit more, because that's a big thing, like the chemicals that are in our like fruits and vegetables, even if like you're trying to like eat healthy, but you're still getting a lot of chemicals that we shouldn't have in our bodies.
Yeah, well, and I remember looking at one of the comments that somebody made. I can't remember who it was, but it was specifically saying about the geographical region, right? And this has a lot to do with, because depending on where you live in the world, you may have access to a specific type of food and you have a different lifestyle, right? If you go to Africa compared to the United States, well, it's a huge difference in terms of the type of food that people ingest, right? And even seasonal for us, although with the globalization, now we can find, 20 years ago, we couldn't find necessarily fruits here in Canada in wintertime. It was always to the same type of fruit, apples, and that was it mainly. but you will not find an orange or a banana. Right now, we have a little bit more of a global expansion. But it is hard now to not be aware of these situations and how we can change some of, not necessarily to progress, but to do better for ourselves, right? Maher, you say that you have read also this paper. Do you have a comment?
Yeah, I found an interesting part, but I wanted to ask about a question regarding this part with recreational drugs, especially cannabis. When it said that the endocannabinoid system, which they found and they isolated from the testes. I know that certain receptors do exist in the testes for these, as well as the brain.
Specifically, yeah.
Yeah, as well as the brain. But are there any other systems in our body that have the same receptors, or it's just confined to these two?
Most likely, we do have more of these receptors in different tissues. I don't know. I have never researched on this specifically.
Because the mental effects of these drugs comes directly from the brain, and that's why they take control of the people after that, especially with dependence in cocaine and heroin. So I know that the receptors do exist in the brain.
Yeah, they do exist in the brain, absolutely.
Yeah, but in regards to other systems in our body, like do these systems or these receptors do exist in other organs or not? This was my...
I believe so. I believe that they should exist in other organs. Amanda, you want to say something?
Yeah, I'm pretty sure the CB1 receptors are found in like a lot of your peripheral organs. So you can even find them in blood cells, like white blood cells, like gastrointestinal tract. I think your pancreas and your endocrine glands, like they're basically found not everywhere, but almost everywhere, like spinal cord, brain.
Yeah, the effects, it will change. Robert, you want to say something?
Yeah, I know the adrenal gland is another big one that has a large number of these receptors.
Yeah,
plays a big role in that.
Okay. I'm sorry, I'm just asking the ones that I see the faces for. They're in my first screen. Does anybody else Raven?
Hello?
Yes.
I was just wondering, everyone's talking about the cigarettes, but I never really asked this question, but is it the nicotine in the cigarettes that causes infertility, or is it just the combination of the thousands of chemicals in the cigarettes? Or
the combination of the other chemicals that are in the cigarettes. Yeah.
I was reading some articles and I was listening to the CBC episode about vaping and infertility, and it was saying it has the same effects. So I'm wondering if it's just nicotine, because vaping does have a reduced amount of chemicals. But
yeah, and that's the problem, right? Because e-cigarettes have been sold more just to advertise that has less nicotine or no nicotine, but has a whole whack of other products that has been proven that can cause damage to your health. So
yeah, vaping is still there's still so many studies going on and unknowns, like they're not regulated like cigarettes, right? So we don't really know the effects that they could be having on the body.
Yeah. Well, we need more studies, of course, and even if all of these products go to the market, and I'm gonna show you one video at the very end for this course that talk about these endocrine disruptors, because that's how we wrap it up the course. And one of the issues with endocrine disruptors in development is that is the additive effect. It's not that 1 + 1 is 2. Is that 1 + 1 + 1 could be 25. Okay. So, and this is one of the issues of us being exposed to so many xenobiotics and to so many endocrine disruptors that we don't know yet how the mix in our body has as an effect. So a lot of these studies are made looking at the specific molecule and the effects on cells. Now, we do, or I hope that you know, that the effect on one cell doesn't necessarily explain what happened in the whole tissue or in the whole body. That's one of the problems with toxicology, right? A lot of these studies are made in cell culture, but those cells are isolated. They are not in the tissue. They are not getting clear in the body. So the levels of toxicity may change when they encounter other compounds that they react in a different way and then create an overall effect in the individual. And that's one of the problems, and particularly during development, that is what compared to us, right? That anything that goes wrong during development can create an effect, a snowball effect, right? So once again, one of the things for this course is that every day that we learn something about everything that can go wrong, we need to be thankful to be alive, because many things could have gone wrong. And yes, maybe we have a little bit of obesity, maybe we have problems with insulin, maybe we have some immune diseases, and maybe we have some heart, but we still made it. We still made it. We are here, and we need to be so thankful of it, despite everything that we can have. Anything else has any other comment regarding these two papers?
I think that another really big contributor, I think it was Kennedy who said that infertility has increased a lot in the last 50 years. So I think people having cancer would probably be a pretty big factor that contributes to this. A lot of people who have cancer are going through radiation or taking chemotherapy drugs, and those drugs are damaging the DNA cells all over the body, including the ones in the reproductive area. So it's stopping cells from reproducing, so it's causing infertility. And a lot of those effects can be reversed after treatments are stopped, but a lot of the people, they can't just stop it when they want, right? They have to do it until their treatment is done. And I think the longer that they do those treatments, the longer that the effects last.
Yeah, and that's what a lot of people that are going to go through a chemotherapy, for instance, they are at the reproductive stage. For males, they're going to preserve the sperm, and for some females, they're going to preserve the eggs, right? So because the chemotherapy is so hard on the body that the effects on those cells may be irreparable, right? Especially if you guys are at the reproductive age, which most of you are at the reproductive age. Now thinking with your loved one, maybe to have a baby, maybe some of you already have a baby. But we gotta think about this. One of the things that I say again and again, getting pregnant need to be planned. Don't leave it to the oopsie, okay? This is one of the things that I always say for this course. Pregnancy needs to be planned. So you need to take the steps, either to change your lifestyle, to eat better, to lose weight, to stop smoking, either cigarette or pot, or take all of these substances that we do sometimes, right? For whatever reason. So medicine that we take also can have some effect. So plan the pregnancies. Don't leave it to the oopsie. Yes, God gave us the blessing of having this baby if you believe in God. But God is not responsible for any of the shit that we do in our life. That's on us, right? So plan pregnancies. Don't leave it to the oopsie. And as we're going to see later when we start organogenesis, everything that can go wrong within the first 4 weeks of that fertilization can go wrong, and we don't even know that we are pregnant. And that's the scary thing. Everything that can go wrong happens within the first 6 weeks of pregnancy. And we don't even know. We sometimes say, oh, I'm a little behind, it doesn't come, and then, oh, phew, I got my period. Well, maybe just too late, right? So, okay, so I hope that you learned something about how to prevent some stuff with these two papers. If that makes you to reflect a little bit on your habits, I'm glad that you achieved that by reading the papers. But as I say, nobody can force you to do anything unless you are willing to do the change. And my point here is just to give you some information that you can use, and if that generates change, great and fantastic. Okay, so Sarah and Amanda.
Yeah.
It's your turn, ladies.
Oh, sure. Did you want me to just do the paper stuff or the report stuff quickly, and then you can go ahead with the pre-lab?
Sure. Yes.
Okay. So I don't want to belabor too much. Just a few comments on the first lab report. I think those marks are out now, hopefully.
Yeah, they are now.
Okay, perfect. Yeah. The big thing, and I like Mary said that she talked about it in lecture 2, is just some instances of plagiarism. So just going to go over it. Hopefully it's review by this point. The biggest thing I noticed and the biggest concern was students copying sentences word for word from the article or the source and then providing a citation. So that is still considered plagiarism. For it to be kind of acceptable, you have to paraphrase everything in your own words and provide the citation. So just kind of to bring that home if you forgot or you didn't know that, but just providing the reference doesn't, or providing the citation doesn't give you the license to copy that. The other thing where I think it kind of becomes a problem with paraphrasing is using your own words and not rearranging the words that the article used. So I see this a lot of the time. Students do this for lists. If an article has a list of effects or list or something, the student will put that list, but just rearrange the order in which the list is. That's still, you still plagiarize that. That's not in your own words. That's not paraphrased. So just be careful for those kind of instances. And then the other big thing I noticed was just citing after every sentence. So that's something that might feel a little awkward, or it looks weird in a paragraph, but really after every single sentence that has an idea or a concept that's not your own, you should be citing, even if it's the same citation over and over and over again. You found a really great paper, that's fine. Use the same citation over and over again, but you can't cite a whole paragraph. So it shouldn't be just one citation right at the end there. But yeah, I don't want to freak people out too much about it. It wasn't a super prevalent thing, just a couple instances, but overall I thought the reports were pretty good. And then if you have any questions about specific marks and that kind of stuff, you can always let us know. Help you there. I don't know if Amanda, if you had anything to add there.
No, I think that was basically it.
Yeah, I would like to add another couple of points. And one is labeling. Right, you are putting the labels. You are changing that into a PDF. Open the file and be sure that your arrows and labels are still where you put them. Okay, we have a couple of instances. I went through all the labs because I do have 3 TAs, so I went through all of the labs very quickly just to see if it was homogeneous, the marking and all that. And I saw a couple of those lab reports where the planaries are here and the labels are over here, or the labels are here and there is no photo of the planaria anywhere to be found. Or the labels were here and the planaria here and the arrows are not connected to anything. Okay, so just be sure that your labels are on. If not, I think somebody just print the photo and label on a paper. They took a photo and then put it up, insert it into the document. Okay, because if we cannot see those arrows and labels on the right position, we can mark, right? So just be careful with that. You put it into a PDF, open it up, double check that the labels are still where they are. When you put a figure, you need a caption. even if we gave you the photo, we need a caption, okay? So don't forget captions, because some of you lost 2, 4, even 5 points because you didn't put captions in the figures. And this lab has a lot of photos that you need to use, okay? What else? There was another thing. Yeah, yes, remember, 10% per day. You submit your lab report an hour later is 1 day. Okay, it's 10% off. And this time we had a person that submitted the lab 8 hours late. So that is 1 day late, okay? Please submit the lab reports into the right folder. Somebody didn't submit the lab report in the date, and then I open up the folder for the lab report from this week that needs to go, and they put it in there, okay? So it happened that I just was looking at who has submitted already, and I opened that. It was like, oh, this is from last week. What is doing in here? Okay. So if you put it in the wrong folder, I'm so sorry, but I am not to check every time just to see if you put it in the right folder. I am opening the folders as it goes, and I close them as it goes. So please just be careful to that, because in this case, the TA was done with the marking, and then I needed to say, hey, could you please go here and mark? This is what, I don't know for you guys, but when I'm done doing my dishes and somebody comes with 3 more balls and say, hey, oh, that's the same feeling, okay? The person said, okay, I'm done. And then all of a sudden said, hey, could you please mark that other one that was submitted in the wrong folder? So just please try to submit it in the right folder, okay? So we can give you the mark back on time. Okay. So, Mandy.
Pre-lab. I can share, right?
You should.
Dr. Martinez, I have a question about the lab that was posted for lab 1.
Can you stay after Amanda gives the pre-lab?
Can everyone see that?
Yes.
Okay, so today's pre-lab is on lab 3, fertilization and cleavage. So in the stages of embryogenesis, this is where we are. We're going over fertilization and cleavage. You guys have already done gametogenesis. So to start off, we're going to look at reproduction and sex. So can somebody tell me what the difference is between these two? What is the definition of sex?
I would say that sex involves exchange in gametes between organisms, or involves more than one gamete from an organism, while reproduction does not necessarily involve multiple organisms. It can be just hydroid dividing.
Yeah, so reproduction is the creation of a new individual, and sex is the combining of the genes from 2 different individuals into new arrangements. So do you think we can have one without the other? Like, can we have sex without reproduction? Okay, well, you can't.
How that process is called? It occurs in euglenas when 2 adult euglenas basically come together, divide their nucleuses into 4 smaller nucleuses, and while 3 of them die, they exchange one and then just leave each other. Yeah, conjugation.
Yeah, so in Paramecium, we can see different types of arrangements that they're capable of. In the Paramecium, we find fission, and this is the reproduction in the absence of sex. So this occurs when organisms are found under optimal conditions, where they can divide themselves to create a whole new organism that's identical to the parent. So that's what we see here. And then there's conditions that are not always optimal for the paramecium. And then this is when they undergo conjugation. So this is sex in the absence of reproduction. So this is what we see here. Oops. So then we have this video that's going to show us fission. Hopefully. What is it doing now?
Amanda?
Yeah.
Go to the top bar on the screen.
Yeah.
Where there is the little dots and share the sound.
Mainly by asexual means. It is a type of reproduction in which there is no form.
We can hear it, it's okay.
It has sound.
And fusion of gametes. In this type of reproduction, a single parent can produce its identical and exact copies. Binary fission in amoeba. It is division of parental body into two nearly equal daughter cells during favorable conditions. In this method, the karyokinesis is followed by cytokinesis. Daughters feed, grow, and repeat the process. Transverse binary fission in paramecium. In this type of fission, the body of paramecium divides transversely. Cytokinesis takes place along transverse axis. Longitudinal binary fission in euglena. In longitudinal binary fission, the body of euglena divides longitudinally. Cytokinesis takes place along longitudinal axis.
Okay, so that was a little video on fission, and then we have another short video on sexual reproduction.
Sexual reproduction in paramecium. Reproduction in paramecium occurs by binary fission and conjugation. Due to repeated binary fissions, paramecium become weak and inactive. To rejuvenate the lost vigor, conjugation takes place. Conjugation. Temporary union between two paramecia of different clones for the exchange of nuclear material and multiplication is called conjugation. It is a type of sexual reproduction. Paramecium have two nuclei, a large macronucleus and small micronucleus. It is responsible for reproduction. Conjugants. Paramecia that undergo conjugation are called conjugants. The two conjugants mating types comes together and closely oppose one another at the region of cytopharynx. The pellicle at the site of the union disintegrates and the cytoplasm of the two conjugants merges with each other is called cytoplasmic bridge. Formation of pronuclei. The macronuclei of both the conjugants disappear slowly. The micronucleus of each conjugant divides meiotically to produce 4 haploid nuclei. Of these 4 haploid daughter nuclei, 3 from each conjugant disintegrate so that 1 nucleus remains in each. The remaining micronucleus in each conjugant divides unequally to form a small migratory or male pronucleus and a large stationary or female pronucleus. Formation of Sincarion. The male pronucleus of one conjugate moves through the cytoplasmic bridge into the other conjugate and fuses with the female pronucleus. It is then called Sincarion or zygote nucleus. After formation of diploid Sincarion, two conjugates are separate. Each separated individual is called X conjugates. The Sincarion of each X conjugate divides thrice by mitosis, so that each X conjugate has 8 nuclei. Of these 8 nuclei, 4 enlarge in size to become macronuclei. The remaining 4 behave as micronuclei. Division of X conjugate. 3 of the 4 micronuclei disintegrate so that each X conjugate contains 4 macronuclei and 1 micronucleus. Each X conjugate divides twice along with its micronucleus and forms 4 daughter paramecia. The daughter paramecium receives 1 macronucleus from the X conjugates, but does not divide along with X conjugate, so that finally daughter paramecium has 1 macronucleus and 1 micronucleus. By the end of conjugation, 8 daughter paramecia are produced from both the conjugates. Significance. The conjugation is a method of nuclear reorganization. It restores the lost vigor and vitality due to repeated binary fissions. The macronucleus of daughter paramecia is formed from Sincarion of X conjugate, so it discharges the physiological activities more vigorously. New characters appear in the daughter paramecia due to exchange of genetic material between conjugates.
Okay. So now we're going to jump into fertilization. So fertilization is the union of 2 haploid gametes to form the single-celled zygote, and it returns to that diploid complement of chromosomes. So this is the same image we showed you last week, and here we see that there's fusion of both the male and female pronucleuses, and that will confirm that fertilization has happened. As you guys saw in lecture, eggs can be divided in different categories based on the amount of yolk as well as the distribution of the yolk. So in terms of yolk quantity, can somebody tell me what microleucethyl means? Like how much yolk is that?
A small quantity?
Yep. And then what about mesoleucethyl?
Probably medium quality, and macro would be a very big quality.
Yeah, exactly. And then for yolk distribution, we have different ways that the yolk can be distributed. Does anyone know what isoleucethyl means? So isoleucethyl is the even distribution of the yolk. And then teloleucethyl is dense yolk throughout most of the cell. And then centroleucethyl is the yolk in the center of the egg.
Yeah, we saw this this week, right? Because we were talking about the movements in fish, amphibians, and birds. And we talk about that mesoleucethyl and teloleucethyl. So we should be able to answer that question.
So then also in this lab, you guys will be looking at frog eggs. So this is a frog egg. We can see the vegetal pole and the animal pole. And how can we distinguish between these 2 poles? If we didn't have these labels, how would we know which one's the animal pole? What's different about the animal pole and the vegetal pole, even just by looking at it?
The black membrane.
Yeah, so the animal pool has this pigmentation. And does anyone know why we would have pigmentation on the animal pool?
Isn't it to protect against UV?
Yeah, exactly. So the pigment is located on the animal pool because that's the side where the embryo is going to develop. And because the eggs are exposed to UV radiation, that pigment will help maintain the integrity of the DNA. And through the different slides and the images that you guys will look at, you're going to be going over different cleavage types. So as explained in lecture, the pattern of cleavage greatly depends on the amount and that distribution of vitellus. So holoblastic and meroblastic, I believe you guys should know. So just, can somebody define what holoblastic means? What type of, like, how does the cleavage furrow form in a holoblastic cleavage? So holoblastic is when the cleavage furrow extends through the entire egg, and then meroblastic is only a portion of the cytoplasm is cleaved by the cleavage furrow. And then there's a lot of other types: equal, unequal, radial, spiral, bilateral, rotational, discoidal, and superficial. So these are going to be important for you guys to identify. When looking at the phylogenetic tree, we can see that deuterostomes have radial cleavage, while protostomes have spiral cleavage. So when looking at the differences between radial versus spiral, on the top here is radial, and we can see that when they undergo radial cleavage, they cleave at right angles to each other, and you get this nice radial appearance. And then for spiral, they cleave at angles that are not right angles, and this causes this spiral arrangement of the cells. And then the cleavage planes, you guys have already seen these in lecture, so if you have any concerns about them, you should really review in your lab manual, but you definitely should understand the cleavage planes. And then in this lab, we're really going to be looking at this transformation from cleavage to the formation of the blastula. So we can see, just based off of this figure, that the appearance is quite different between the sea urchin, the frog, blastula formation, and then human. So through this lab, you guys are going to be learning about that formation of the blastula. I think that is it. Do you have anything else to add, Mary?
Just and share the screen so you can see everybody. Okay, yeah. No, that's it. So you have the notes already. You have the photos for this lab already available, and you also have, for those that don't have the book yet, you also have the PDF of the chapters in the book. Remember, you gotta get the book. I cannot continue giving this available because of the copyright. We are just saying this for help those that haven't received the book. Okay.
I received it yesterday.
Good, Maher. Yeah, so, and that's what I said, that I will be sharing those until you get the books, but you have to get the books, or ask somebody else to send you copies or something. That I can't, as a faculty member, I cannot continue to violate the rules. So read the chapters, answer the questions as specified, okay? Remember, the more complete your answers are, the more chances you have to be hitting the jackpot, that you put the right words that we are looking for, okay? We are sometimes very lenient into, okay, have the keywords that we want, yes, no. Again, I want to repeat this for I don't know which time. Derivation means the origin of the word, whether it's Greek or Latin. It's just not the definition of the word. I want to say side means did, goat means this, okay? Blastula means this. This is how we cut the word, and this is what it means in Greek or in Latin. that is a derivation of the word, because I have replied, I think right now, like to 5 emails to this specific question. And last week I mentioned this during the lab too, okay? It's the root, the derivation is the origin of the root, what this means. And it's so helpful. I wish that you guys here need to take Greek and Roman derivations, because I did have to do it in my university, and that helped me so much through my biology degree, because I just can, oh yeah, this is this and this is that, and then it explains it all so much easier, right? So use the photos, label well the photos, double check that the labels are in the right section. Paraphrase so you don't incur again into plagiarism, okay? And yeah, so just if you can see whatever you had from last lab that can help for this lab, or the last lab is completely different as of the rest of the labs from now on, okay? But on the label inside and how to argument your answers, that might help, okay? And remember, we don't need an introduction, objectives, materials, and methods. We don't need any of that, okay? Just go straight, follow the same headings as subheadings from now on. This is really important to use those headings as they are in the notes, so that TAs know exactly your answers to what question. Does anybody else have any question? If you had, you can stay in the room. If not, try to enjoy your weekends. If there is a little bit of snow that fall, at least here. So if you are an avid of skiing or skating, maybe that's a good activity, because with the skis, we maintain social distancing anyways. So that's a good thing. Just try to get a little bit of time for yourself, half an hour, just meditation. That sometimes help. And I know that you're gonna need this, because next week, many of you are on midterms and things like that. And for that reason, you don't have a paper for next week. So I didn't put up any paper, so you're gonna have a little bit of a, okay. I know that you enjoy very much reading the papers that I put up, but I give you 1 week of respite, so you can focus on those courses that do have midterms. And if you haven't finished your assignment that you had since the very beginning, maybe this weekend will be a good time. It's the video. I'm talking about the video assignment that is due on Friday next week. Okay, and I have already sent some feedbacks to some people that have shared their videos with me and say, okay, just do this, just that. Remember, for those that haven't submitted the video, review it, change it into PDF, into MP4, okay, and listen to it. If it sounds too fast for you, you're gonna sound fast for me, okay? I had, no, you will not, I haven't had time. This is just going so crazy. On feedback on the outline, okay? I thought that I will have time, but I haven't had any time at all. So I don't want to promise something that I won't be able to do, okay? And I thought that this morning I will have time to go and find a video for you guys. I'm gonna try to do it after and post it on D2L2. I just need to go back, but I don't keep copies of the videos that my past students have made, because otherwise my computer will explode in terms of how much I can have, okay? So I'm gonna go into my Google Drive to see if I have something that I can share with you guys in terms of a good, but if just listen to your voice, if it can be here. Just be sure that the transitions between the slides are okay. There is not any space where you have a black slide, but you are talking like for 5 minutes. If you are talking for more than 40 seconds in one slide, maybe think about splitting that into 2 or 3 slides, okay? Don't put a whole whack of images that is like this big. It's better just to use a big image that is clear, that is not pixelated, okay? Another thing, use the pointer. Okay, if you put an image, you see me pointing every time that I'm using, that I'm teaching you, right? So use a pointer. You put an image, as you can see here, and you're talking about something in the image, and I stop paying attention into what you're saying because I'm trying to locate that part. Okay, if you are using a pathway, just follow with your mouse. As you can see here and here and here, so they go, okay, so I can focus. But if you tell me, as you can see here, and you are talking, I'm trying to find where is the part that you are saying that I should be paying attention to. Okay, so use the mouse. It doesn't bite, as you know by now. So use the mouse. Okay. Okay, so that's it. If you don't have any questions, you can leave. And if you have some questions, Helen, I received your email and it's funny. So you can stay and have the questions that you have, okay? Other than that, enjoy your weekend as you can.
Thank you. I was just wondering, how do we cite the